Hello, class. I wanted to make a short video about macromolecules. We spoke about these guys previously, and I want to make sure that you're comfortable with this material. So, I wanted to kind of like do a really brief overview, some things that are important as far as the test is concerned, and some recognition. A lot of the test issues that we have with this portion of material is the identification. Most people know like the simple properties and features of different types of macromolecules, but the aspect of like how these guys actually look, that's probably one of the biggest problems that we have. So, without further ado, I want to look at carbohydrates real quick. And carbohydrates, better known as sugars or starches for us, these guys, as mentioned earlier, have the elements carbon, hydrogen, and oxygen, and they're inside of a 1 to 2 to 1 ratio. A good example will be glucose. Glucose is C6H12O6. And glucose still follows this pattern. If I reduce the 6 to 12 into 6, that's that 1 to 2 to 1 ratio. What's important about this is that the carbon and the oxygen molecules in glucose will allow it to form this hexagonal shape that we can see here. And as a result, we'll get a single hexagonal shaped molecule. Now, what's generally not drawn inside most of the images that you see are that there are other elements that are there, like you see some H's and OH's that are present. Um, and then you have something that looks like that. Now, each of these little corners right here would represent where carbon would be at as well inside this connection. If you want to do the time and say like, trying to figure out how these guys were like the C6H12O6 and actually count oxygens and carbons, you'll be able to figure that out. Well, what happens also with these guys is that if you're going to connect two of these guys, what will happen is that this oxygen at the end will be used to actually form a bond with another molecule. And I'm not going to draw all the H's. So the first situation that we had, which was simply this situation right here, this guy is known as a monomer. Now, carbohydrates have a suffix that's associated with them, and that suffix is known as a saccharide. So we will define this as a monosaccharide. Okay. Now, when I get two of these guys together, as the one we just had earlier, the prefix for two is di. And as a result, I'll call this a disaccharide. And if I had three or more bonded together, I use the prefix poly. Poly means many, so there'd be a polysaccharide. So an example of a monosaccharide that we spoke about earlier will be glucose. This is not actually very sweet if you taste it. A disaccharide would be sucrose. Another good disaccharide would be a lactose that's found inside milk. And then polysaccharides would be complex carbohydrates. And that includes things like starches. Okay. Remembering the fact that carbohydrates are quick energy sources, the big highlight that I want you to get from it is just simply looking at the actual structures of these. So, if for test purposes, if you're able to see that you had one of these, or two of these, or three of these, you'll be able to identify and say, okay, that's a carbohydrate. And I identify carbohydrates as substances that have sources of quick energy. Um, and that part is important. They're a good source of energy, and part of the reason why is because if you remember the simple cellular respiration equation that takes place inside the mitochondria, you start off with glucose, C6H12O6, and then you add that to 12 moles of oxygen, and you create 6 moles of oxygen, 6 moles of carbon dioxide plus water, and then the most important thing was ATP. So, with this entire aspect, this carbohydrate is the thing that's needed in order for us to make the most important thing for our life. In that case, it's going to be ATP, which is our energy source. That's an article on It's Learning that talks about the importance of ATP and how ATP works. You're free to look at that on your own. The next one I wanted to look at were proteins. And I wanted to look at proteins for a particular reason because I wanted you to see how proteins kind of really align themselves. Proteins themselves are complex within themselves. The actual protein itself is the polysaccharide or the mini. Okay, we'll say these guys are polypeptides. And these polypeptides are held by polypeptide bonds, which are bonds that bond the monomer of proteins together. And the monomer of a protein is an amino acid. Just like the picture before, I don't want you to get lost on the fact of how these things look. So an amino acid will look like this, in which you have nitrogen with some hydrogen stuck to it, carbon, hydrogen, carbon, okay? And that carbon is going to be double bonded to a O, then to a OH, and then you're going to have what we call an R group. All right, some important parts of this amino acid. So that's what this is, an amino acid. And the important parts will be these guys right here. This area right here is known as a carboxylic acid. This is the acid part of it. And then this area right here, this NH2 that you can see here, this is known as an amine group. So these are functional groups that we'll talk about like in biochemistry and organic chemistry. The other thing that's very important is looking at this R group, and there's a whole bunch of different types of R groups that can be present. Obviously, the most simplistic would be hydrogen, okay? But these guys can have cyclic structures and things of that sort. What you need to know is that all amino acids look the same, except the thing that changes are the R groups. And there are 20 different types of R groups, which makes it where there's 20 different amino acids. Proteins, what's their function? They make up structures. They have functions within the body. When we look at each other, we're generally looking at the structures that are created from the proteins, and those things are important for us inside this class as far as knowing them. The amino acids and the proteins are also digested by us inside meats and nuts and things of that sort. In that case, we digest the protein, break it down to its amino acids, and then rebuild them to create human stuff. I mentioned this part in class. I don't want to just kind of get on the soapbox over that, but if you have any questions, you can ask me about that during class as well. The last thing I want to look at, I'm kind of skipping lipids, and the reason why I'm skipping lipids is deals with the fact that I thought like this was the easier one, and I'm going to probably go back to it later. And but prior to that, I'm going to look at nucleic acids and nucleotides. Nucleic acids, there's only two of these guys that we use and talk about in this class, and that's DNA and RNA. Deoxyribonucleic acid and ribonucleic acid. The monomer for these guys is known as a nucleotide. And a nucleotide within itself is made up of its own parts. One part that is made up of is something that we've seen before, okay? In this case, this thing right here is a sugar. The sugar is attached to a base. The difference between this base is that this base has nitrogen, So as a result, this is known as a nitrogen base. And then the last thing is that sugar is also attached to, I'm going to make it a P and just put P sir O. This group right here is actually a phosphate group. If you've taken chemistry, this is a polyatomic ion that's PO4. It has a -3 oxidation number for it. So it's the same one that you talk about in class. But these are the monomers. Being able to see this and identify it is very important. The other thing that's important about DNA is DNA has A, T, and C and G as far as the nitrogen bases are concerned. And then RNA does not have T. You replace thymine with uracil. So it's going to be A, U, G, and C. So that part is important. DNA does not leave the nucleus. RNA has the ability to leave the nucleus, and they serve for obviously genetic material. We spoke about the aspects of being able to break these guys down and how they work. Um, they're important. You can actually get nucleic acids even in diet, but obviously the big part of it is that it contains our genetic material, contains all the information about the organisms that's based from genetics. Not to mention, obviously, the big aspect of the influence of environment on how an organism survives or looks, or appearance, and things of that sort. That's also important. The last thing that we had were lipids, and I didn't want, like I told you, I did not want to bore you down with lipids, but it has two parts to it. It has a glycerol molecule, and that molecule looks like this. And then after that, it has a chain of hydrocarbons. And we spoke about this in class. What's important as I finish drawing this chain of hydrocarbons is that we have to ask the question, does each carbon on the chain of hydrocarbons, does it have the maximum amount of hydrogens? Or is it a situation in which uh, that hydrogen, that carbon only has like one hydrogen there? If so, it makes it where, just like the image that I'm drawing here, it makes it where there's a kink inside the chain. This right here is the difference between what we call a saturated or unsaturated uh, fat or lipid. This will also take place over here. I'm going to probably stop this chain a little earlier just for time's sake. But in this case, what's important is that there's no double bonds on this one. Okay, this one there's no double bonds, means it has the maximum amount of hydrogens connected to each carbon. This is saturated with hydrogen, so we say this is saturated fat or saturated lipid. However, with this one, it does not have the maximum amount of hydrogens because of the double bonds that are present. These guys that are saturated at room temperature, okay, they're usually solids. Okay, this is usually the case for lipids that are animal fats or animal fats that we talk about. It's like if you have lard or it's like bacon grease, as it gets cooler during room temperature, it will start to solidify. While this one right here is what we call unsaturated. And you can really see these guys at room temperature, they're going to be liquids. And the other part about these guys that's important is that most of the time you can find these guys in plants and things of that sort. It makes up like your olive oils and things of that sort. The other big part that's important is that lipids make up many important things inside the human body, such as cholesterol. We have what we call LDL lipids and HDL lipids. And, those are types of cholesterol. But then on top of that, they make up every membrane inside the human body for that double phospholipid bilayer, or what we call the fluid um, model, the mosaic fluid model of the membrane. But it allows for the fluidity movement of the membrane. And the other part that's important is that association with carbohydrates allows for connections between different cells and signaling inside the organism and things of that sort. That's a really important part for us as well. So I gave you all these guys in a nutshell. I tried to make this as fast as possible. You can always go back and look at these. Also, if you have any problems, you can always contact me. You can send a message via um, It's Learning itself, or we also have a Remind. So it's important that you have registered for that Remind. If you can't remember how to do that, you can also look at the It's Learning on the syllabus at the very beginning in the first unit, and it will give you the information at the very top of the syllabus for the Remind 101. And if you can't find your syllabus, that copy is on It's Learning. It's really the first thing that we did inside this course. So thanks for your time.